Let's talk now about the liver. And the liver is part of the accessory organs of the digestive system. If you remember, we said the accessory organs are organs that the food will not pass through them, but they will secrete enzymes and other stuff that will help in digestion. And we said we have the liver, the gallbladder, and the pancreas. All of them are accessory organs. And we will start with the liver. The liver is made out of layers of cells. Those are called the hepatocytes. The hepatocytes are technically performing the function of the liver, which is filtration. So it will filter and detoxify, detoxification of the materials that coming from the digestive system. So those hepatocytes are arranged in lobules, like in hexagons. At the center of each lobule, there is something we'll call the central vein. It's a central vein that is draining blood, that is collecting blood from the sides of the lobules. And this blood is drained from something called portal venule. So there is blood coming to the portal venule. From where this blood is coming from the portal vein. And this blood in the portal vein is coming from where? It's coming from the hepatic portal. And if you remember, what puts the blood in the hepatic portal is the superior and inferior mesenteric veins. Also, we have the gastric, the splenic, and the pancreatic veins. All of those are putting blood into the hepatic portal, and from there it goes to the liver. Remember, the superior and mesenteric, they are coming from the small and large intestine, right? And this blood, although deoxygenated, but it's filled with nutrients, so it's nutritious blood. And those nutrients are coming from outside the body, right? From the food. So we need to make sure that they are safe to go into the circulation, because once they are in the circulation, technically they can go, for example, to the brain or to the kidneys, right? Or to the lungs. And if there are toxins, they will cause problems. Therefore, we need to make sure that whatever we absorb, whatever we ate is fine to be in the general circulation. Therefore, it has to go to the liver first, and the liver will do the filtration and detoxification. So the small and large intestine will return the blood in the superior and inferior mesenteric vein. With other veins, they will put in the hepatic portal. Hepatic portal goes into the liver, and from there into the portal veins, and from the portal veins it will go to the portal venule, and from the portal venule that blood would be processed through something called cystinoids. Those are like small canals between the hepatocytes. So as the blood moving here, it will get filtered and detoxified. And from there, it will go to the central vein. And from the central vein, it goes to the interlobular vein. And from there, into the hepatic vein and into the inferior vena cava. So this is the circulation of blood through the liver. All of this. And you need to be familiar with it, right? So that's how the blood ended up in the central vein. And now we have deoxygenated blood, nutritious blood. However, it is detoxified blood as well. So this blood now is safe to go into the general circulation. Beside the portal venule, we have a portal arteriole. This is bringing oxygen to the liver cells, to the hepatocytes. And we have the bile duct. This bile duct is technically collecting bile from the cells, producing it and sending it to the gallbladder, and we will talk about this. But those three together, they are a triad, a triad or three venules, and those are on the sides of the lobules. Before moving to the gallbladder, I want to show you this. So here, this is the central vein. This is the portal venule, right? So the blood coming here. This is the portal arteriole, also blood coming here. And we have the hepatocytes. Also, we have there the citellate cells. Those are the ones responsible for producing the bile. But also we have Kupffer cells. Those Kupffer cells are macrophages, immune cells, and those will deal with any bacteria that might escape from the large intestine. So the Kupffer cells or the spider cells, those cells you find them in the liver to deal with any bacteria or microbes. But the bile will be secreted and goes back to the gallbladder. Let's go back and talk now about the gallbladder. And the gallbladder, its main function is to store the bile. But what is the bile? Basically, think of it like a group of chemical structures, steroid type or steroid-based chemical structures. This is the main component of the bile. There is also water in the bile. There is the bilirubin, if you remember it, from the blood cells. That's responsible for the colors of the feces later, right? But mainly there is, and of course there are salts, inorganic salts. But the main thing here is this steroid-based chemical structure. What does it do? It will emulsify fat. fat. What does it mean emulsify fat? If you remember, fats are hydrophobic, which means they are afraid from water. And if they are hydrophobic, it means we can't absorb them. Therefore, we need to put them in a sac. That is, this sac is hydrophilic. This sac is hydrophilic, and therefore we can absorb them. So this sac is the bile. This sac is the bile. So the gallbladder stores this bile. And as we saw, we have cells in the liver that produce this bile. This bile will be drained through the bile ducts, right? So we will have all of that bile ducts put their secretions into the left and right hepatic ducts. This hepatic duct will form the common hepatic duct, okay? The gallbladder, the secretions of the gallbladder will go through the cystic duct and together will form the common bile, okay? So we have, for the arrangement of the bile, we have left and right hepatic ducts that will give us the common hepatic duct. This common hepatic duct will merge or will open to the, or will be merged with the cystic duct, and this is coming from the gallbladder, and this will give us the common bile duct. This common bile duct, where it will go, eventually it will go through the pancreas into duodenum, the first part of the small intestine, okay? And that is where the bile will get secreted into the small intestine. As we said earlier, the bile will emulsify fat. Another function of the bile is to reduce acidity of the chyme. Remember, the chyme is coming from the from the stomach, which has a pH of 3, okay, which is a little bit acidic for the intestinal cells, for the duodenum cells. Therefore, we will mix it with the bile, and the bile will increase the pH. The bile and the pancreatic juice will increase the pH of the chyme. So that is for the liver. Now this bile would later get reabsorbed and restored in the gallbladder. Okay, so there is like a bile circulation or enterohepatic circulation. This is the, how we reabsorb the bile salt. I think around 93% or 94% of the bile is being reabsorbed. Okay, and again, remember, it will emulsify the fat. Sometimes we will get, for the gallbladder and the liver in general, sometimes we will have gallstones, and those are, solidified materials from the bile salt. Okay, usually it's cholesterol and bilirubin will get solidified and will accumulate, and sometimes will block the cystic duct. And if it block it, it means this sometimes will cause inflammation, pain, and so on. And usually they will try to destroy them, but usually they will go ahead and take the gallbladder out. So they will do a surgery and to cut the gallbladder out. So because there will be inflammation there. And in this case, this gallbladder or this patient will live without gallbladder, and they have to be careful with their diet because they cannot eat or digest plenty of fat. Then for the pancreas we already saw it in the endocrine system. If you remember, we said it has it's a dual organ. It has the endocrine function and the exocrine function, right? Endocrine is the insulin, glucagon, right? And the exocrine is what we'll see today, is the digestive enzymes. So there in the pancreas, we said we have the pancreatic islets, right? Or Langerhans islets. So those will secrete the insulin and glucagon. But between them, or actually the majority of the pancreas, is Achnar cells. So those Achnar cells are exocrine glands. They will secrete the pancreatic juice or pancreatic enzymes into the pancreatic duct, which will take them out to the duodenum. What do we have in the pancreatic juice? What do we have? We have a mix of enzymes, okay, digestive enzymes. Like what? Like amylase, for example, for sugar, like a trypsinogen for proteins, for sugars or carbohydrates, okay, trypsinogen, chymotrypsinogen, another enzyme, also for proteins. And now remember the gen, trypsinogen, chymotrypsinogen should remind you with pepsinogen as well. Those we said that those are the inactive form of the enzymes. We produce them inactive because we don't want them to eat or consume those cells. They will get activated in the duodenum. In the duodenum, the trypsinogen will turn into trypsin, and chymotrypsinogen will turn into chymotrypsin. And they will, again, break down the proteins. Also, what do we have there? We have, in the pancreatic juice, we have also the bicarbonate, okay? The pancreatic juice will contain sodium bicarbonate, which will be broken down into sodium ions and bicarbonate ion. And this bicarbonate ion is extremely important because it will also increase the pH of the chyme. As we said earlier, we have HCl from the stomach, from parietal cells of the stomach, produce HCl, and we will get us chloride ion and the hydrogen ion. The bicarbonate will bind the hydrogen ion, and therefore it will reduce the acidity of the chyme, and this is critical. So that is for the pancreas. I'll stop here. For the last video, we'll talk about the specific types of digestion happening in the digestive system.